heard my voice on somebody else's speaker, and I think maybe Chris's speaker or something next door
Okay.
since I wasn't muted.
Okay. All right.
Okay,
So let me know when you want me to get started. I'm ready to go.
great. Get started, please.
Okay. All right. Well, thanks, Jane. As the title implies, my talk today is going to include not only research, some of the research that I did at the institute where I was located, and research that involved collaboration with both students and scientists, but also my experiences in sharing outreach and teaching strategies with the instructors there in marine biology and marine geology. And I'll also mention some of the other outreach types of activities that I was involved with that include educational outreach. So my pathway to the Institute for Marine and Antarctic Studies at the University of Tasmania, where I did my year-long sabbatical, was a result of my interest in being in Tasmania because I have family there, but also because the University of Tasmania's IMAS, the Institute for Marine and Antarctic Studies, is ranked in the top 2 research institutions in the world for Antarctic research, which I have an interest in and expertise in. And also they're ranked in the top 10 for marine science, which of course is my focus, my research focus. So just to remind everyone where Tasmania is located, it's about 8,000 miles southwest of California. It is an island state just off the southern coast of Australia. And here's Tasmania relative to Sydney and Melbourne on the mainland. And to the right there, you can see an elevation map that shows that in the center of Tasmania, there's some mountainous regions with some beautiful lakes and surrounded by coastline, most of which is sparsely populated. I was located in Hobart, which is the largest city in Tasmania. It has about 200,000 people or so, and it's located on the western side of the Derwent estuary, and I'll talk more about that estuary in just a little bit. So this is the view from the institute where I worked. This is Hobart, the wharf at Hobart. Here's another view from the institute, and this one also includes the view of the mountain, Mount Wellington, which is now known as Kunyanyi. And this is where I had my lunch every day. This is a view looking back toward the institute from the wharf. And here's a picture of the institute itself, where the lunch area was, is just at the top of that glass area that you see there in front. So again, IMAS is part of the University of Tasmania, and they conduct a wide variety of research, primarily focused on Antarctic and marine science research, but they also do a lot of teaching there as well as part of the University of Tasmania. This is the view from my desk. I took pictures almost every day because things were changing out there. There'd be a new suite of sailboats or yachts or cruise liners or seaplanes coming in, as this is a touristy spot, and also is quite a spot for sailboats and yachts. In fact, it's the final end of the Sydney to Hobart yacht race, which is a very famous and well-known yacht race around the world. So I also included my microscope by my desk so I could be continually distracted by that great view out of my window. So here's a picture of Hobart from Mount Wellington, from the top of Mount Wellington. And that little red area that you can see just to the right of center, that's their brand new Antarctic ship, and that's located not too far away from where IMAS is located. So you can see the Derwent there with the Tasman Bridge crossing the Derwent. This is a view north, looking at the Derwent estuary as it winds its way into the heart of Tasmania, and eventually it turns into the Derwent River. And one of the reasons why I'm showing you the Derwent is because it was a focus of one of the projects that I worked on with both students and scientists at the Institute for Marine and Antarctic Studies. My interest was in the contamination of the Derwent. And the primary source of contamination is this zinc works, which is located just north of Hobart. And this zinc works has been in place since 1916, generating a good portion of the world's zinc. And as a result, it's also been contributing for most of those years to the contamination, the heavy metal contamination of the Derwent. And this is one of the things that I wanted to have a look at. So even though they've cleaned up their act a little bit these past few years, there's still a lingering legacy of contamination as a result of the zinc smelter and other contaminators as well, including a paper mill, but primarily the zinc smelter generating zinc contamination, for example, in the Derwent. You can see on the left there, we've got concentrations of zinc in the water. And then on the right, these are zinc concentrations in various organisms, including some fish and crayfish, for example, abalone. And these are well above, in most cases, the acceptable limit for zinc in these creatures as food anyway. And especially you note the crayfish and abalone, as well as the eel concentrations. Those are higher in all probability because they tend to hang around near the sediment or in the sediment. And that's where a lot of these contaminants are sequestered and emanating from. So the objectives of this particular study that I'm going to be talking about today was, first of all, to work with UTAS undergraduate students and the scientists to measure some of these contaminants, both in the environment and in the creatures that I work with, which I'll talk about in a little bit. And to do this, we sent students out to collect sediment cores from 3 of the bays on a transect south of the zinc works, and then made measurements of the sediments and the interstitial water, that is the water in between the grains called pore water, that included zinc concentrations, and eventually, hopefully, will include some other contaminants, particularly heavy metals as well. My focus was on the examination and identification of living populations of foraminifera and ostracods, and I'll explain what those are in a bit, from these samples, with the idea that if we can calibrate the amount of the contaminants incorporated in these organisms and compare that with the concentrations in their environment, then if we can get a consistent calibration with different species do different things, then we could then apply that to the fossil record as well and get a history of the bioavailability of these contaminants through time. And what do I mean by bioavailability? Well, that basically refers to the percentage of the contaminants that are available that are incorporated or can be incorporated into the organisms. So, for example, if there's asbestos in the glue holding the tiles at our feet together, that's not bioavailable to us, hopefully. But if we were to grind up the entire building and measure the amount of asbestos, we might see there'd be quite a bit of asbestos in the building. But again, it's not bioavailable, so it's not as much of a concern. And this happens also in the natural world, where particularly in the sediments, some of these contaminants can get bound up and not be available to organisms. And so it's important to try to differentiate between what's available and what isn't. And by definition, if you can find these contaminants within the organism itself, then that's bioavailable. And the creatures that I'm going to be talking about, want to be focusing on, that again I'll describe here in just a little bit, are tiny. And so we can get lots of them in a small sample. We don't have to try to capture fish or collect abalone or these larger organisms. And they may help us to assess not only in the modern realm what's going on in the recovery of these ecosystems from contamination in various places along the estuary, but also give us a history by looking at the fossil record to see how we're doing in terms of recovery from the contamination, how much it's still being incorporated into these organisms. So this is a picture of the Derwent showing where the zinc smelter is located relative to the 3 blue arrows where we had a student go out and collect cores from Newtown Bay, Cornelian Bay, and Montague Bay on a south transect away from the zinc smelter. This is a picture of Montague Bay. So you can see Hobart there in the distance across the Derwent. And these inset pictures basically are showing the student as he collected cores from the 3 bays, including Montague Bay, from a ship that is used on a regular basis to monitor the amount of contamination in zinc in particular in the water and other larger organisms. And we're trying to advocate for the idea that we might be able to look at some of these smaller organisms and do this more efficiently and effectively and to get a better idea of how these contaminants are affecting the ecosystems. So this is us in the lab. This is on the left there, myself helping Noah, the tall undergraduate, help process those sediment cores. The picture in the center is Xana Chase. She was the chemical oceanographer, paleoceanographer that I worked with on a number of projects, including this one. And then to the right is Noah, again, making measurements of some of these sediments, in this case, looking at oxygen concentrations or profiles of oxygen concentrations. This is another undergraduate, one of the 4 that I've been working with. She's processing core samples, sieving them. She also, along with 2 other undergraduate students, looked and picked out foraminifera and ostracods from some of these samples as well. And they're continuing to do that beyond just the time that I was there. Okay, so what are foraminifera? This is the group that I tend to focus on for my research. They are single-celled organisms, and there are ones that float around in the open ocean near the surface of the water, and they're called planktonic. And then there are benthics, those that live on or within the seafloor sediments at every water depth, from ankle deep to the deepest depths of the ocean. And typically I focus on the deep water ones, but for this particular project, I'm focusing on shallow water foraminifera. And so many of them have an internal shell that we call a test. And you can see some of the pictures there of some of the varieties of them. They are very sensitive, both biogeochemically, ecologically, and morphologically, to changes in their environment, which makes them very valuable as tools to assess how the oceans have changed through time, how the environment has changed through time, and ultimately actually how climate has changed through time. They're used extensively for lots of different projects. And we're only now really applying their capabilities, these creatures, as a tool to assess contaminants, to assess pollutants. So here's another picture showing a wide variety of these different types of foraminifera. These are about the size of a grain of sand. They come in a wide variety of sizes, but most of them are about the size of a grain of sand. To distinguish the living from the dead, I treat the samples with rose Bengal, which basically stains the protoplasm of these little creatures red, so I can distinguish which ones were alive at the time of collection and which ones weren't, and thereby be able to assess living populations and choose some of these for our living analyses. Again, these creatures are single cell. It's kind of like having an amoeba in a shell, although again, technically these are internal shells, so we call them tests. It took me quite a bit of time to figure out the taxonomy of these shallow water groups. It's kind of a hodgepodge in many respects, and there's a lot of variety even within a species. There's a wide variety of shapes, and especially since some of these shapes can be influenced by contaminants. So even within the same genera, which these two illustrations are, you can see they're quite different. Here's some scanning electron microscope pictures of these taxa. And again, you can see there's, even within the same genus, there's a lot of variability in there. And there's a lot of what biologists and paleontologists would call ecophenotypic variation, which means that within the same species, you can get a lot of variety in the morphologies in different types of environments, which makes identifying species really difficult, but it's critical that we do that because not only do we want to understand what's going on in terms of the populations, but also we want to look at the biogeochemistry of these species, and different species do different things. So again, this project's kind of based on the idea that the biogeochemistry of the tests of the foraminifera reflect ambient conditions. So some of these creatures, and the ones that I focused on, make their test out of calcium carbonate, which is the same materials that a snail or a clam make their shells out of. And these creatures incorporate chemicals, the chemical composition, I guess I should say, of the tests of the calcium carbonate is influenced by their environment, including the concentrations of some of these contaminants. And using new technologies, we're able to figure out the biogeochemistry of single tests. And again, remember, these are the size of a grain of sand. So these are some scanning electron microscope photographs of 3 different species. Now, these aren't the same specimens from the study that I'm talking about. We haven't done those analyses on these yet. These are from a study that I conducted in Lake Macquarie, just north of Sydney, where we found some of the same species in the Derwent samples that we looked at. So that's encouraging, and we're going to be able to compare these data. But basically, what we use a laser system at the Australian National University in Canberra, Australia, and basically ablate a hole in specific spots along in the test, in the shell of these creatures. And again, remember, this is the size of a grain of sand, so we're able to really precisely place these holes, and each one of these holes generates hundreds of individual measurements of the concentrations of different trace metals. And so from these, and we can average these, and we can see what the variations are within the test itself to get a better handle on the concentrations of various contaminants, in this case, zinc, calcium, or zinc, and we use a ratio zinc calcium to make sure that we can compare from one species and one group to another. So these again are data from Lake Macquarie, and I'm in the process of working on a paper to publish this material. But so this is 2 different species, Spirulina vivipara and Quincoloculina seminula, and looking at the lead calcium in each of the tests of these organisms, and comparing that with the gray dots here, which is the concentrations of lead in the sediments that they were living in. And you can see just visually that it seems to be a pretty good match. And if we look at one of those species, then we can see that with Quincoloculina seminula, that there's a pretty good relationship there between the lead in this particular species and the lead in the sediment that it is being exposed to. We also looked at ostracods, which are little crustaceans. And again, these are about the size of a grain of sand as well. They're like a little shrimp that's encased in a carapace that looks very much like a shell. And these are also very sensitive to environmental changes. And my taxonomy of ostracods is pretty rusty, so I were partnering with an ostracod researcher in Melbourne. And so when I go back in January, we'll be sending our ostracods off to him for identification. He's very excited because neither the foraminifera nor the ostracods have ever been looked at in the Derwent estuary. And so there's a lot of potential interesting aspects about both the ostracods and the foraminifera in terms of their distributions within the estuary. Okay, so I could go on and on about different research projects that I'm involved with, including the one I just was talking about, but I want to make sure that I left time to talk about other aspects of Australia, including some of the creatures that I encountered. So on my walk in to the bus that I took every morning into work, I would frequently see beautiful, colorful birds along the way, including the pink one there, the galah, and the eastern rosella, the one on the right with the red head and colorful body, and also bandicoots up in the right left. So that kind of looks like a rat and it's about the right size, but these little guys are hopping around, and I'll talk a little bit more about them a bit later on. But I saw those on Maria Island, along with wombats there in the center, and the forest kangaroos, which can be quite rare. They're not abundant everywhere. They're only found in certain places. So it was great for me to be able to run across the mob of them, as well as some of the other creatures as well that I encountered while I was in Australia. So I wanted to mention that there's a lot of things that you have to get used to in Australia. And although they speak English, it's Australian English, and there's a lot of different words that mean different things or that are totally unique to Australia. And so this was a sale to support the local elementary school. And so they weren't selling condoms for $1. Rubbers in Australian means erasers. So, and there's lots of these kind of language pitfalls that you can fall into if you're not careful. Okay, so while I was there, I interacted with the instructors, both in terms of oceanography, biology, and geology. And I was invited out to Maria Island, which is just off the coast of Tasmania, for this week-long course that marine biologists teach to undergraduates. And I'm going to talk about that a little bit. The first time that I went out there, I was just observing the course that for a day, this week-long course where undergraduates are involved in different aspects of research, marine biology research, during that time, and then they use those data to come back and work on them back in Hobart. The second time that I went out there, though, I was invited out to help teach this course, this marine biology course, to high school students that had come from all over Australia. So IMAS has the equivalent of our dual credit program, only in this case, they bring the high school students to Hobart and then eventually to Maria Island, where they spend a week out there doing the same sorts of projects that the undergraduates did. And these high school students get college credit for this. And it was a pretty impressive program. And so I want to talk a little bit about that. So this is a view from Maria Island. It has lots of different types of beaches. This is another view where the students conducted some research that I'll talk about in just a bit. This is called Painted Cliffs. And there were lots of fossils, although the students didn't really work on these for research. They piqued my interest. I was quite interested in the variety of fossils that I found on the island. And so this is another beach, and the island has quite a number of wombats. So this is Mother Wombat and her baby out for a stroll on the beach late in the afternoon on Maria Island. And this is Maria Island is a reserve now. It used to be a penal colony, and then it was used for agriculture, and then it was used as a cement factory. But now it's a reserve, and it's managed by the Aboriginal group that whose ancestors originally occupied the island. So they manage the island and make sure that its environments and wildlife are well taken care of. So they limit the number of tourists, but nevertheless, it's a very popular tourist area. There's only a few places to stay, though, so most people that come there are obligated to stay in tents, and there's no vehicles except for the ranger that's there. You have to bring a bike or walk around the island. And as a result, these wombats, at least on this section of the island, are relatively calm and used to people. So I got relatively close to this wombat and her baby. And as I mentioned, this was a penal colony. So where we stayed was this historic penal colony that's been kind of renovated there. But the bricks that you can see there, those were all made by convicts. And the structures were originally made for convicts that were held on this island. And as you can see, the wombats are pretty comfortable. This one's taking a snooze there by the building where we all stayed. So one of the research projects that's done on the island as part of this course is marine diving, and they measure environmental parameters, but a lot of what they do are fish counting in the reefs along the island, the kelp reefs, the temperate kelp reefs, which are a major focus for research at IMAS. And the data that the students collect are helpful for those research projects as well. So what happens is these students, some of the students will be diving, others will be doing some of these other activities that I mentioned, and then they'll all switch. So everybody gets a chance to do the diving and the fish counting, and everybody gets a chance to do these other activities as well, and they all pool the data, and then different groups will are obligated then to give a presentation about the data at the end as part of their assessment, their grade, essentially. What's really interesting is they've been doing this for over 10 years, and they've been trying to keep the methodology the same. So the data that they're getting this year can be compared to 10 years ago, and so students are able to look at the patterns. So it's not just doing it in isolation. You're able to look to see where your data fit on this long-term pattern, which is pretty cool. So this is those divers. They're in this case measuring the oxygen of some of the environments where they were that they surveyed. This is another group that was looking at some of the creatures that are living in this estuary. So they just collected material from a net tow in various spots in the estuary, and looking at how the creatures are as the salinity changes in this small estuary. And this was the instructors setting out this long tape study area where students are going to then make assessments of the changes in organisms from low tide to high tide mark. And this is what the students are doing. So they have these metal squares that there are distributed randomly along that line that was just set up. And within that square, then they identify and count all of the different creatures in there, and then they can multiply that up to get an idea of the abundances and the changes that occur from low tide to high tide. And they did this in this area, which is the fossil cliff area. They also did it in the painted cliff area. And so I was helping with all of these in terms of making sure the students were made identifications correctly and set up the transects correctly, et cetera, along with the other instructors. But my main focus was out on the small boat, where we collected both plankton toes and grab samples of various places off the coast of Maria Island. And once we had those samples, then we would take them back and look at them and identify the creatures in the microscope. And I put both of these pictures on there because these students are working night and day. They would get up at 6 o'clock, and some of them then would be going out doing diving at that time. Then they come back, and others would be doing other things like the transects. Then they would work on the samples or the data, and especially the microscope samples. That took a long time. So they were working well into the evening every night. After dinner, we would give lectures or presentations about different aspects of marine science. And so I gave a few of those lectures, as well as helping each day with the microscope identifications of these creatures. And then again, at the end, they're giving these presentations. They would go back to Hobart at IMAS, and they gave presentations to the public. So the public was involved or invited. And so their families would get a chance to see them give a presentation. And some of these families are flying in from a long way off on the mainland to see their son or daughter give these research presentations and earn their college credit grade. While I was there in Hobart, also lots and lots of people were coming through. So postdocs and visiting scientists from all over the world, Europe, Japan, the United States, South America, as well as Australia. And one of the Australians that came by while I was there was a paleo-oceanographer from the University of Queensland. And when she found out that I was there, she invited me to help them teach this marine geoscience masterclass in Brisbane at the University of Queensland. So this masterclass focuses on getting students into the idea of using these large ocean drilling program ships to generate major international research. And so this was an international group of students that had applied and been selected to participate in this particular masterclass. So they flew me up to Brisbane, where I spent a week helping teach this masterclass. So this is one of the classrooms where we got a presentation from, in this case, the International Drilling Program coordinator for Australia and New Zealand, telling us about the capabilities of the ship, etc., which the students would then eventually use that information for a proposal that I'll talk about in just a little bit. I was primarily in charge of helping with the processing, how to process samples and how to take samples from cores. This is Helen Bockstock, the paleoceanographer that invited me up, who was in charge of the organization of the masterclass, and some of these international students. And I gave lectures and also helped with the identification of microfossils. So that was part of this course in identifying and figuring out. We gave them tasks to figure out where these samples might have been from in the world based on the characteristics of the microfauna microfossils that they were encountering. We are also the Queensland Institute of Technology is just down the river. We had a beautiful couple of beautiful trips on a ferry down the river to visit Queensland Institute of Technology. And there we were given demonstrations and hands-on exercises in how to use AI for identifications, for taxonomic identifications of some microfossils, which was pretty interesting. But also we were given presentations by this group at this institute that work on with JPL in the United States that work on the Mars rover. And so they had a lot of really cool, interesting things, insights about what they did, and they designed one of the pieces of the Mars rover. So they have communication and have influence on what the Mars rover does and how it can do it. And we were given virtual tours also of Mars, which was pretty cool. So it wasn't all just the oceanography. We were exposed to some of these other aspects that these universities are being involved with. So at the end, the students broke up into groups, and they would put a proposal together that would answer a burning scientific question. And so these students spent a lot of time in discussion figuring out what question to answer and how they could answer it and if it could be answered. And of course, us as instructors would go by in each group and help them decide and help them with the process of putting a proposal together, which was pretty cool. Okay, so now I want to talk a little bit about some of the outreach activities that I was involved with. And that includes one that I gave at Hobart Brewing Company. That sounds a little strange, perhaps, but there's a long-running program where the IMAS, people at IMAS, and also people at the CSIRO, which is a government agency focused primarily on fisheries, right next to IMAS. Some of the people from there are involved with this program, as well as the Antarctic Division, which is just down the road. So postdocs or graduate students volunteer, and they basically get speakers every month to give a talk at this Beer Aquatic. And this is based on the idea that, on the premise of the society presentations that used to be given in England. So in England, they used to have, way back when, you'd give a presentation at the Royal Society, and there'd be everybody from the public and experts as well in the audience, and it would be primarily kind of a discussion. And of course, that was before PowerPoint. And so the rule is that you can't use PowerPoint in this presentation. You can bring a few props if you want, but it's going to be just you talking, and then discussion and answering questions with the audience. I loved it. It was a blast. I really enjoyed doing that, and there was a lot of people there. So it was standing room only. There was about 100 people there, and I'm sure they were there for the beer more than what I had to say. But nevertheless, it was really interesting and fun, and I went to other ones as well. So this is well attended by the community, and not just students and not just people from IMAS, but also just the community have a great interest in what's going on in terms of the science and what's going on in their environment. So it was pretty cool. I also gave a presentation in the Island of Ideas public talk series. This is organized by the University of Tasmania, and it's advertised around Australia. And again, it's another one of those regular series of talks that are organized where they talk about topics that are of interest to the community, not just the scientists, and it's very well attended. So there was about 350 people or so at this particular talk, and another, I don't know, 165, I guess, online. And again, this time I could use PowerPoint, so I did. And that was also really a great experience. So being able to interact with people from the community, answering their questions, and representing not only CSUB, but also representing the University of Tasmania. So it was fantastic. Now, there's too many things to talk about here of all of the things that I got a chance to do. But one of them was to get a tour of their brand new icebreaker, their Antarctic research ship. I had sailed on the predecessor to this one, but this one is huge and brand new. It had only been to the Antarctic a few times when I toured it. And So researchers at IMAS went out of their way to make sure, because they don't give tours, they only give previews essentially of people that have labs or whatever are going out on the ship. And so with the help of researchers, I was able to get on this ship and see this amazing ship, and also then talk with the researchers that are going to be taking cores off this ship, because it's not really set up really well for that. So we were kind of trying to strategize as to how best to do that. So lots of these kinds of things happened to me and were available to me as a result of collaborations at the University of Tasmania at IMAS. So I gave a number of talks. I also gave talks at the fisheries conference. They were interested in the aspect of contaminants in the estuary, and gave research talks at IMAS, as well as talks on experiential learning there as well. So at this stage, I want to just advocate for a year-long sabbatical. You get more than just twice as much as the semester long. It took a semester before people even realized that I was going to be around for a while and what I did, even though I gave talks, etc. And it really, the second half of the sabbatical was really one where a lot of opportunities came into play that I wouldn't have had had it only been a single semester. And again, also that the sabbatical leaves, when you get a chance to do them, to get out and away and meet new people, collaborate with new people, and explore, even take your family along. These places are just amazing, and there's lots of opportunities, not just in terms of research, but teaching and outreach that you can potentially learn from, like I did. So at this point, I guess I'll